Hello, this is Miss Dagenford, and I wanted to continue our lesson on the cell by going over the nucleus and then something called the endomembrane system. Now, I know you were asked about Build-A-Bear, and I know some of you might be thinking that is an odd question to be asked. But I want you to imagine the goal of Build-A-Bear is not only to build a bear, but also to accessorize the bear. If you were ever there, or even if you know what it is, you know that once you build your bear, after you stuff it and get everything inside that you want, the heart or the sounds, but you then have to choose out an outfit, you put on different hats, and then you get the bear and they put it in a little box for you, which I do think is kind of clever. And then you carry it out. So it gets delivered. So think about Build-A-Bear as being an assembly line. And if you have that in your mind, you'll understand the goal of the nucleus and then the endomembrane system. Now, the nucleus itself, let's take a look at that. The nucleus is going to contain three distinct regions. The chromatin, which is, I said in the earlier video, is the DNA. It basically contains the code for making proteins. Now, proteins, remember, were organic molecules that did a variety of things. So, antibodies, enzymes, structural proteins like keratin, collagen, your cornea, your bones. So they make us up, but then they're also the workers in our body. So proteins are incredibly important. So DNA would be the code for making those proteins. So they go hand in hand. In fact, two ways that you can basically kill a cell is you either by disabling the protein making machinery, antibiotics can do that, or you can disable the DNA making factories, and some chemotherapy drugs can basically disrupt DNA replication. So, very crucial jobs here, containing the code for making proteins and then making proteins. So, what's this all about? Well, like I said, the chromatin would be the instructions, the DNA. We have the nucleolus, which is going to make the ribosomes, and then we have the nuclear envelope, which is the interface between the cytoplasm and the nucleus. It's the boundary. It's basically the membrane that keeps the nucleus and keeps everything from mixing. And then you'll notice the rough ER is going to be coming out from the second membrane of the nucleus, and it has ribosomes on it. So you can see this is kind of the intricate relationship here. So you've got nucleus containing the chromatin, the nucleolus, and then the nuclear envelope. So what in the world is the big deal about this endomembrane system? Well, we need something to make the protein, but we also then have to deliver it. We have to sort the protein. You have to modify proteins, just like, if you walked into a Build-A-Bear, you see like 50 kids all making different types of bears. Well, think about what each kid is doing is making some are making bunnies, some are making unicorns, some are making teddy bears. I even had somebody say they made a Darth Vader Build-A-Bear. And so then you have to sort them all and put them in the little boxes. So just like that, every cell in your body is making different proteins, and a lot of them. You're making upwards of thousands of different proteins in your cells. And so you need a way of not only making them, but then sorting through them, modifying them, and then getting them to a place where they need to be. And so there are going to be several organelles that are going to be part of this. Now, the actual membranes that are involved would be the rough ER, Golgi, lysosomes. But other organelles interact. Remember, the nucleus contains instructions for making them. The ribosomes are going to be the part that actually makes the protein. And then you have vesicles, little membrane sacs that are going to um, transport these proteins. So again, just like Build-A-Bear, you're going to build a protein. And so remember, the idea is that we have a little assembly line. So let's go through this assembly line. The nucleus is going to contain instructions to make a protein. The ribosomes are going to build the protein. They're going to attach amino acids together and they're going to make the proteins. Thousands of different proteins. And remember, you've got upwards of 50,000 ribosomes in a cell, if not more. A bacteria cell can have 15,000. So our cells easily have way more. So ribosomes build proteins, and then the rough ER and Golgi body are both going to modify the proteins. We can add different molecules to them. We can make them fold properly. And remember, proteins have to fold a specific way. So these organelles can help the protein do that. But then once you have all the proteins made, you have to kind of sort through them. And think of this as like a UPS or an Amazon delivery. So the Golgi body is going to be this. It's going to finish. It's going to provide the finishing touches. It's going to modify the protein, and then it's going to sort them out and then send them different places. And it's going to deliver the proteins to where they need to go. So for instance, here are a couple of different examples. We have the plasma membrane that makes blood types or cell signaling proteins. So we can make proteins for the membrane. Your blood types are blood types because they're different types of proteins. We can make proteins that go outside the cell, like intestinal enzymes. We can make antibodies that go outside the cell into your bloodstream. Or we can deliver the proteins to other organelles, like lysosomes, that contain powerful enzymes. So let me um, wrap up and show you a case study. In this case, cystic fibrosis. Now, in this case, this is the normal membrane protein. And so let's follow through. We've got the gene for the normal CFTR, cystic fibrosis transmembrane conductance regulator. So we have the gene for it, and then that gene is going to be made into, or it's going to be, the gene is going to be used to make a protein, CFTR protein, in the ribosome, and then the rough ER is going to modify it. The Golgi body is going to modify it, and then it's going to be transported to the membrane. And so in this case, this is a normal CFTR, but if there are problems, then you have cystic fibrosis. And then just to wrap up, we have lysosomes, which again, they contain very powerful enzymes that can break things down. They come from the Golgi body. And then the smooth ER is not making proteins, but it's making lipids like steroids. But they also make enzymes that break down toxins and break down um, things that might be destructive. So smooth ER is going to be the next organ that we do. So that gives you a sense of how the cell is basically a protein manufacturing factory.